Hey everyone, welcome to Advanced Exercise Physiology, Chapter 3, Bioenergetics. The objectives of this chapter are 1. To discuss the functions of the cell membrane, nucleus, and mitochondria. 2. To define the following terms: endergonic reactions, exergonic reactions, coupled reactions, and bioenergetics. 3. To describe the role of enzymes as catalysts in cellular chemical reactions. 4. To list and discuss the nutrients that are used as fuels during exercise. And 5. To identify high-energy phosphates. 6. To discuss the biochemical pathways involved in anaerobic ATP production. 7. To discuss aerobic production of ATP. 8. Describe the general scheme used to regulate metabolic pathways involved in bioenergetics. 9. To discuss the interaction between aerobic and anaerobic ATP production during exercise. And 10. To identify the enzymes that are considered rate-limiting in glycolysis and the Krebs cycle. For those of you who are taking notes, here is a simple outline that you can follow with major topics involving cell structure, biological energy transformation, fuels for exercise, high-energy phosphates, bioenergetics, the aerobic ATP tally, efficiency of oxidative phosphorylation, the control of bioenergetics, and the interaction between aerobic and anaerobic ATP production. To start with an introduction, metabolism is the sum of all chemical reactions that occur in the body. Anabolic reactions synthesize molecules, while catabolic reactions break down molecules. Now, bioenergetics is converting foodstuffs such as fats, proteins, and carbohydrates into energy. Now, when we look at the cell structure, we can first see the cell membrane. This is a semi-permeable membrane that separates the cell from extracellular environments. Within the cell, there is a nucleus, which contains genes that regulate protein synthesis. This would be referred to as molecular biology. And also, within the cell, you have cytoplasm, the fluid portion of the cell, which contains organelles such as mitochondria. Looking at the diagram of a muscle cell and its major organelles, you can see that it's made up of the cellular membrane, the cytosol, nucleus, mitochondria, as well as myofilaments. So in summary, metabolism is defined as the total of all cellular reactions that occur in the body. This includes both the synthesis of molecules and the breakdown of molecules. Cell structure includes the 3 following major parts: cell membrane, nucleus, and cytoplasm, called sarcoplasm in a muscle. The cell membrane provides a protective barrier between the interior of the cell and the extracellular fluid. Genes, which are located within the nucleus, regulate protein synthesis within the cell, and the cytoplasm is the fluid portion of the cell that contains numerous organelles such as mitochondria. Now, looking at molecular biology and exercise science, molecular biology is the study of the molecular structures and events underlying biological process. Ultimately, it's the relationship between genes and cellular characteristics that they control. Genes code for specific cellular proteins, which is the process of protein synthesis. Now, with exercise science, exercise training results in modifications of protein synthesis. Strength training in particular results in an increased synthesis of muscle contractile protein. Molecular biology provides tools for the understanding of the cellular response to exercise. Now, if we look at the overlapping fields of study in molecular biology, we have biochemistry, physiology, genetics, which all lead to the study of cellular function, proteins, and genes. Now, if we look at the biological energy transformation, and in particular cellular chemical reactions, we have endergonic reactions. These require energies to be added, and they're called endothermic. Exergonic reactions are ones where energy is released, and they are referred to as exothermic. If we look at coupled reactions, There's a liberation of energy in an exergonic reaction which drives an endergonic reaction. In particular, if we're looking at the biological energy transformation of the breakdown of glucose, this is an exergonic reaction. Within coupled reactions, again, the energy given off by the exergonic reaction powers the endergonic reaction. Now, focusing on oxidation and reduction reactions, you have oxidation, which is the removal of an electron, and reduction, which is the addition of an electron. Oxidation and reduction are always coupled reactions. They often involve the transfer of hydrogen atoms rather than free electrons. Hydrogen atoms contain one electron. A molecule that loses a hydrogen also loses an electron and therefore is oxidized. Now, if we look at NAD and FAD, NAD is nicotinamide adenosine dinucleotide, which is oxidized from NAD plus and reduced from NADH. FAD is referred to as flavin adenine dinucleotide, which is oxidized from FAD and reduced from FADH2. Both play an important role in transfer of electrons. Looking more closely at the structural formulas for NAD+, NADH, FAD, and FADH2, when NAD reacts with two hydrogens, it binds to one of them and accepts the electron from the other to form NADH. Conversely, when FAD reacts with two hydrogens, it binds both of them to form FADH2. Now, moving on to enzymes. Enzymes are catalysts that regulate the speed of reactions. They lower the energy of activation. The factors that regulate enzyme activity include temperature and pH, and ultimately, enzymes interact with specific substrates in a lock and key model. Now again, looking at how enzymes catalyze reactions, the enzyme lowers the energy of activation for both a non-catalyzed reaction and a catalyzed reaction. Now, looking more specifically at the lock and key model of enzyme action, you have a substrate such as sucrose that approaches the active site on the enzyme. The substrate fits into the active site, forming an enzyme-substrate complex. Ultimately, the enzyme releases the products glucose and fructose. Now, the diagnostic value of measuring enzyme activity in the blood refers to damaged cells that release enzymes into the blood. The enzymes level in the blood indicates disease and/or tissue damage. The diagnostic application is that elevated lactate dehydrogenase or creatine kinase in the blood may indicate a myocardial infarction. This is just one example of a diagnostic application. Further examples of the diagnostic value of the enzymes in the blood are listed in the table below. Now, if we look at the classification of enzymes, almost all enzyme names end in ASE, such as kinase, which adds a phosphate group, dehydrogenases, which remove hydrogen atoms, oxidases, which catalyze oxygen reduction reactions involving oxygen, and isomerases, which are involved in the rearrangement of the structure of molecules. Factors that ultimately alter enzyme activity, like we mentioned, are temperature. Small rises in body temperature actually increase enzyme activity. Exercise results in an increase in body temperature, so exercise can directly alter the enzyme activity by raising the body temperature. pH also alters enzyme activity. The changes in pH reduce enzyme activity. Lactic acid production during exercise is another example of how exercise can alter enzyme activity. Now, if we look a little bit more closely at the effect of body temperature on enzyme activity in the graph below, we can see that within the normal body temperature range, slight increases in body temperature actually increase activity. But there's a certain point where if the body temperature rises too high, especially during exercise, that the body temperature can actually lower the enzyme activity. And looking a little bit more closely at the effect of pH on enzyme activity, you can see that both an increase and a decrease in pH can affect enzyme activity outside the optimal pH range for that enzyme activity within the body. This would include the example of lactic acid building up during exercise, ultimately decreasing enzyme activity. So in summary, enzymes serve as catalysts to regulate the speed of chemical reactions. Enzymes are classified into categories based upon the type of reaction that the enzyme performs. And two important factors that regulate enzyme activity are temperature and pH. Individual enzymes have an optimal temperature and pH which they are most active. Now, looking at the types of fuel for exercise, we're going to discuss carbohydrates. Carbohydrates would include monosaccharides, disaccharides, and polysaccharides. Glucose is the most well-known and is a typical blood sugar. Glycogen is the storage form of glucose in the liver and muscle. It's synthesized by the enzyme glycogen synthase, while glycolysis is the actual breakdown of glycogen into glucose. Now, moving on to fats as a fuel for exercise, we have fatty acids, which are the primary type of fat used by the muscle. There are triglycerides, which is the storage form of fat in the muscle and adipose tissue. Triglycerides are broken down into glycerol and fatty acids via lipolysis. Phospholipids are actually not used as an energy source. And what might pertain to exercise is that steroids are derived from cholesterol and they're needed to synthesize sex hormones. Now finally, we will discuss proteins. Proteins are composed of amino acids. Some can be converted to glucose actually in the liver. This is referred to as gluconeogenesis. Others can be converted to metabolic intermediates, which can contribute as fuel in the muscle. Overall, though, protein is not a primary energy source during exercise. So in summary, the body uses carbohydrate, fat, and protein nutrients consumed daily to provide the necessary energy to maintain cellular activities both at rest and during exercise. During exercise, the primary nutrients used for energy are fats and carbohydrates, with protein contributing a relatively small amount of the total energy used. Glucose is stored in animal cells as a polysaccharide called glycogen, and fatty acids are the primary form of fat used as an energy source in cells. Fatty acids are stored as triglycerides in muscle and fat cells. Proteins are composed of amino acids, and 20 different amino acids are required to form the various proteins contained in cells. The use of protein as an energy source requires that cellular proteins be broken down to amino acids. I now want to move our discussion to high-energy phosphates. The most common is adenosine triphosphate, also called ATP. ATP consists of an adenine, a ribose, and 3 linked phosphates. The synthesis looks like ADP plus a phosphate giving you ATP, where the breakdown you have ATP utilizing the enzyme ATPase that will lead to ADP plus a phosphate as well as energy. Please see the diagram below to see the structure of ATP. Now, the model for ATP as a universal energy donor is also depicted in the diagram below. Energetics, we first want to discuss the formation of ATP. This can happen through phosphocreatine breakdown, the degradation of glucose and glycogen through glycolysis, and the oxidative formation of ATP. Now, through anaerobic pathways, ones that do not involve oxygen, you have PC breakdown and glycolysis. But there are also aerobic pathways which require oxygen, and these are referred to oxidative phosphorylation. Looking at the anaerobic ATP production, you have again the ATP PT system, which is an immediate source of ATP. But you also have glycolysis, where glucose gives you 2 pyruvic acids or 2 lactate acids. The energy investment phase requires 2 ATP, but the energy generation phase produces 4 ATP, 2 NADH, and 2 pyruvate or 2 lactate. Now the question should be asked, does creatine supplementation improve exercise performance? Now the depletion of PC may limit short-term high-intensity exercises. Creatine monohydrate supplementation can increase muscle PC stores. Some studies show improved performance in short-term high-intensity exercise. However, inconsistent results may be due to water retention and weight gain. But supplementation can increase strength and fat-free mass with resistance training. Creatine supplementation does not appear to pose health risk. However, there may be variability in the supplement purity. Now asking another practical question, are lactic acid and lactate the same thing? Or can the terms lactic acid and lactate be used interchangeably? Lactate is actually the conjugate base of lactic acid. Lactic acid is produced in glycolysis, which rapidly dissociates to lactate and H+. So the ionization of lactic acid forms the conjugate base called lactate. So the terms actually can't be used interchangeably because they are actually different things. Now looking at the diagram below, we can see the 2 phases of glycolysis. We have an energy investment phase and an energy generation phase. Below is a diagram of glycolysis during the energy investment phase. Again, energy investment refers to the ATP being invested into the process. However, you can see during glycolysis, there's a far more detailed energy generation phase, and the energy generation would now be producing the ATP that were initially invested into the glucose during the process. Now, if we take a look at hydrogen and electron carrier molecules, they transport hydrogens and associated electrons. They are able to transport them into the mitochondria for ATP generation in an aerobic process, and they're able to convert pyruvic acid to lactic acid in an anaerobic process. We have both nicotinamide adenine dinucleotide, again NAD, as well as flavin adenine dinucleotide, FAD. The NADH is shuttled into the mitochondria. NADH produced in glycolysis must be converted back to NAD. This is done by converting pyruvic acid to lactic acid and by shuttling hydrogen into the mitochondria. A specific transport system shuttles the hydrogen across the mitochondrial membrane located in the mitochondrial membrane. Below you can see a conversion of pyruvic acid to lactic acid, where the addition of 2 hydrogens to pyruvic acid forms NAD and lactic acid. So in summary, the immediate source of energy for muscular contraction is the high-energy phosphate ATP. ATP is degraded via the enzyme ATPase as follows. The formation of ATP without the use of oxygen is termed anaerobic metabolism. In contrast, the production of ATP using oxygen as the final electron acceptor is referred to as aerobic metabolism. The muscle cells can produce ATP by any one or a combination of 3 metabolic pathways. You have the ATP PT system, you have glycolysis, and you have oxidative formation of ATP. The ATP PC system and glycolysis are 2 anaerobic metabolic pathways that are capable of producing ATP without oxygen. Now when we look at aerobic ATP production, this is going to involve the Krebs cycle, also referred to as the citric acid cycle. In this cycle, pyruvic acid is converted to acetyl-CoA, in which carbon dioxide is given off. The acetyl-CoA combines with oxaloacetate to form citrate. Citrate is metabolized to oxaloacetate, and 2 carbon dioxide molecules are given off. This produces 3 molecules of NADH and 1 FADH2. It also produces 1 molecule of GTP, which ultimately produces 1 ATP. The diagram below can show the 3 stages of the oxidative phosphorylation. Here is a further detailed diagram of the Krebs cycle. Now looking at fats and proteins in aerobic metabolism, first we have fats, which again can be classified as triglycerides, which give us glycerol and fatty acids, and also just straight fatty acids, which lead to acetyl-CoA during beta oxidation. Glycerol is not an important muscle fuel during exercise, however. Moving on to protein, protein can be broken down into amino acids and then converted to glucose, pyruvic acid, acetyl-CoA, and other Krebs cycle intermediates. Please see the diagram below to look at the relationship between the metabolism of the proteins, carbohydrates, and fats, which we have discussed in the Krebs cycle. Now looking a little bit more at the aerobic ATP production, this actually takes place in the electron transport chain. Oxidative phosphorylation occurs in the mitochondria. Electrons removed from NADH and FADH are passed along a series of carriers, cytochromes, to produce the ATP. Each NADH produces 2.5 ATP, while each FADH produces 1.5 ATP. This is called the chemiosmotic hypothesis. Hydrogen protons from NADH and FADH are accepted by O2 to form water. Now looking closer at the chemiosmotic hypothesis of ATP formation, the electron transport chain results in a pumping of hydrogen proton ions across an inner mitochondrial membrane. This results in a hydrogen proton gradient across the membrane. Ultimately, energy is released to form ATP as hydrogen ions diffuse back across the membrane. This is an example of a diagram representing the electron transport chain. Now looking at beta oxidation is the process of converting fatty acids to acetyl-CoA. In order to get fatty acids, we must first break down triglycerides. Once we have the fatty acids, they can be converted to acetyl-CoA, which can be ultimately used as fuel. Acetyl-CoA enters the Krebs cycle and is ultimately used as energy. Here is a diagram representing beta oxidation. So in summary, oxidative phosphorylation, or aerobic ATP production, occurs in the mitochondria as a result of a complex interaction between the Krebs cycle and the electron transport chain. The primary role of the Krebs cycle is to complete the oxidation of substrates and to form NADH and FADH to enter the electron transport chain. The end result of the electron transport chain is the formation of ATP in water. Water is formed by oxygen-accepting electrons. Hence, the reason we breathe oxygen is to be used as the final acceptor of electrons in aerobic metabolism. Now looking at free radicals are formed in the mitochondria. Free radicals are produced by the passage of electrons along the electron transport chain. Free radicals can react with other molecules in the cell. However, this damages the molecule combining with the radical. Aerobic exercise promotes the production of free radicals in the mitochondria. This is not due to oxidative phosphorylation in the mitochondria, however. Looking at the aerobic ATP tally per glucose molecule, you can ultimately see that the NADH and the FADH that are produced during the Krebs cycle lead to the majority of the ATP created during oxidative phosphorylation utilizing the electron transport chain. Now, if you take a closer look at the ATP balance sheet from before, historically, one glucose actually produced 38 ATP, but recent research indicates that one glucose only produces 32 ATP. The energy provided by NADH and FADH are also used to transport ATP out of the mitochondria. The three hydrogens must pass through the hydrogen channels to produce one ATP, and another hydrogen is needed to move the ATP across the mitochondrial membrane. So if we're looking at the efficiency of oxidative phosphorylation, 1 mole of ATP has the energy yield of 7.3 kilocalories. 32 moles of ATP are formed from 1 mole of glucose. So the potential energy released from 1 mole of glucose is 686 kilocalories per mole. The overall efficiency of aerobic respiration is 34%, where 66% of the energy released as heat. So in summary, the aerobic metabolism of one molecule of glucose results in the production of 32 ATP molecules, whereas the aerobic yield for glycogen breakdown is 33 ATP. Now, the overall efficiency of aerobic respiration is approximately 34%, with the remaining 66% of energy being released as heat. Now, if we look at the control of bioenergetics, we have rate-limiting enzymes, which is an enzyme that regulates the rate of the metabolic pathway. The modulators of these rate-limiting enzymes are levels of ATP and ADP plus phosphate. When you have high levels of ATP, it actually inhibits ATP production. However, if there are lower levels of ATP and high levels of ADP plus phosphate, that will stimulate ATP production. Calcium may also stimulate aerobic ATP production. Below you will see a diagram that represents an example of a rate-limiting enzyme. In addition, here is a table that will show you a variety of rate-limiting enzymes of metabolic pathways. So in summary, metabolism is regulated by enzymatic activity. An enzyme that regulates a metabolic pathway is termed the rate-limiting enzyme. The rate-limiting enzyme for glycolysis is phosphofructokinase, while the rate-limiting enzymes for the Krebs cycle and the electron transport chain are isocitrate dehydrogenase and cytochrome oxidase, respectively. In general, cellular levels of ATP and ADP plus phosphate regulate the rate of metabolic pathways involved in the production of ATP. High levels of ATP inhibit further ATP production, while low levels of ATP and high levels of ADP plus phosphate stimulate ATP production. Evidence also exists that calcium may stimulate aerobic energy metabolism. Finally, looking at the interaction between aerobic and anaerobic ATP production, the energy to perform exercise typically comes from an interaction between both the aerobic and anaerobic pathways. This will ultimately depend on the duration and the intensity of the exercise. For short-term, high-intensity activities, there's a greater contribution of anaerobic energy systems. However, for long-term and lower to moderate intensity activity, the majority of the ATP produced comes from aerobic sources. Now this graph below and chart will look at the contribution of aerobic and anaerobic ATP production during sporting events. In summary, the energy to perform exercise comes from an interaction of anaerobic and aerobic pathways. In general, the shorter the activity, i.e. the higher the intensity, the greater the contribution of anaerobic energy production. In contrast, long-term activities, which typically are lower to moderate intensity, utilize ATP produced from aerobic sources. Now, with all the information covered, I want to give you some study questions to help you learn the information in a little bit more detail. Question 1. Please list and briefly discuss the function of the three major components of the cell structure. Question 2. Briefly explain the concept of coupled reactions. 3. Define the following terms: bioenergetics, inorganic reactions, and exergonic reactions. Question 4. Discuss the role of enzymes as catalysts. What is meant by the expression energy of activation? Where do glycolysis, the Krebs cycle, and oxidative phosphorylation take place in the cell? 6. Define the terms glycogen, glycogenolysis, and glycolysis. 7. What are high-energy phosphates? Please explain the statement that ATP is a universal energy donor. 8. Define the terms aerobic and anaerobic. 9. Briefly discuss the function of glycolysis in bioenergetics. What role does NAD+ play in glycolysis? 10. Discuss the operation of the Krebs cycle on the electron transport chain in the aerobic production of ATP. What is the function of NAD+ and FAD in these pathways? And 11. What is the efficiency of the aerobic degradation of glucose? 12. What is the role of oxygen in aerobic metabolism? 13. What are the rate-limiting enzymes for the following metabolic pathways: ATP-PC system, glycolysis, Krebs cycle, and the electron transport chain? Please briefly discuss the interaction of aerobic versus anaerobic ATP production during exercise. Question 15. Discuss the chemiosmotic theory of ATP production. 16. Briefly discuss the impact of changes in both temperature and pH on enzyme activity. And 17. Discuss the relationship between lactic acid and lactate. This concludes Chapter 3 on bioenergetics. Please refer to the lecture and your textbook if you have any questions, or feel free to email me at any time. Thanks.